ever stop to think, like really think, about how a pill or a liquid medicine, whatever you or someone you know might take, how it always seems to be just the right dose?
Yeah.
Not too strong, not too weak, and it like works the same way every single time.
Right, right.
But how do we actually know that's going to happen? Like, how do they make sure?
So that's a great question.
Today, we are diving deep into a world that honestly, most folks probably don't think too much about, but is so crucial. Analytical methods in formulation testing. You know, those tests and checks that scientists do to make sure every single dose of medicine is exactly what it should be.
Hmm.
Now, looking at all the stuff you sent over, especially that transcript, what was it called? 99 Analytical Methods in Formulation Testing. I think it was from season 7, episode 9.
Yeah, that's the one.
It's obvious this whole area is like the bedrock of pharmaceutical quality. So our mission in this deep dive is to break it all down, you know, make it clear how these methods work, but more importantly, why they matter so much for the medicines we depend on. Okay, let's unpack this.
Absolutely. So to start with, we got to understand that analytical methods are really the foundation for making sure any drug, like whether it's a pill, a capsule, an injection, whatever, has the right identity. We need to be 100% sure what that substance actually is.
Right.
And then, of course, its strength, or what we call potency, which basically means the exact amount of the active ingredient in it. And, of course, there's the whole quality thing. Does it meet all the standards? Is it pure? Meaning, does it have any, nasty contaminants in it?
Makes sense.
So we'll be looking at some of the key techniques that scientists use, both when a new drug hits the market for the first time and then, how they keep checking it over time to make sure it stays stable and effective.
Okay.
And a really important part of this whole thing is, of course, the industry standards. Those super strict rules and the expectations set by those regulatory bodies like the FDA, they're the watchdogs, making sure drug companies are doing everything right to keep patients safe.
So basically, we're talking about the science that makes sure every medicine you pick up from the pharmacy is safe and it actually does what it's supposed to do.
Precisely.
And by the end of this deep dive, hopefully, everyone listening will get a much better grasp on, the hows and the whys behind drug quality and why all these processes are in place to protect us as patients.
Hmm.
So let's kick things off with the big question. Why are these analytical methods so essential?
Well, it all boils down to quality and performance.
Okay.
Making sure that every single batch of a drug that's made,
Mm.
every single one, consistently meets the exact specifications.
Yep.
It's not enough to just be kind of close. Each unit, every tablet, every milliliter has to be right on the money.
So it's about total consistency. Like no matter when you buy that medicine, whether it's today or a year from now, that first pill is going to be identical to the last one.
Exactly. And there are several pieces to this. First, there's verifying the amount of the active ingredient, the stuff that actually makes the medicine work.
Mm-hmm.
Like if a bottle says 500 milligrams, you better believe scientists are making sure there's exactly 500 milligrams in there, plus or minus a tiny, tiny bit.
Got it.
Then there's the checking for anything that shouldn't be there. Things that might form as the drug gets older or maybe got in there accidentally during manufacturing. We call those degradation products and impurities.
Ah, so like making sure there's nothing harmful lurking in there, right?
Right. Or if something is there that it's, below a safe level. And the third part is making sure the physical stuff is right too. So like if it's a cream, does it spread easily? If it's a pill, is it hard enough to swallow, but will it also dissolve properly in your body? That's called dissolution testing.
Interesting. So it's not just about the chemicals themselves, but how the medicine is actually put together, its form, and how that might affect how it works in the body.
Yeah, absolutely. All this testing is basically to guarantee that a drug will do its job properly, throughout its whole shelf life. Because if it breaks down too quickly or it doesn't dissolve properly, it's not going to work as well, right?
Makes perfect sense. So when does all this testing actually happen? I'm guessing it's not just like one big test at the end.
No, you're right. It's a multi-stage process. One critical stage is what we call product release testing. So these are the tests that are done on every single batch of a drug before it can leave the factory and, end up on the pharmacy shelves.
Okay.
Think of it as the final quality control check, like the last hurdle before it gets to the patients.
The gatekeeper.
Exactly.
Mm.
So this is where they confirm that the product meets all those standards we talked about: identity, strength, quality, and purity.
And what kind of tools these scientists use for these tests? I know the transcript we have for this episode doesn't go into a ton of detail about the specifics, but I've seen some of the other materials you shared, and it seems like they've got a pretty amazing arsenal of techniques.
Oh, absolutely. Yeah, you're right. The transcript might not list every single method, but there are some key ones that are used all the time in pharmaceutical analysis.
Okay.
Chromatography, for example, is a big one.
Chromatography.
Yeah, especially techniques like HPLC, which stands for high performance liquid chromatography, and GC, which is gas chromatography. Basically, these methods separate out all the different molecules in a sample, like think of it as sorting them by size or some other property.
Yeah.
And that lets us identify and measure them.
Wow.
HPLC is particularly amazing because it can separate really complex mixtures, like think about all this stuff in a pill. It has incredible resolution, which means we can spot even tiny, tiny amounts of impurities.
Wow.
GC is especially helpful for those volatile compounds, the ones that easily turn into a gas.
Huh. Interesting.
And then there's spectroscopy.
Spectrum?
Yeah, like UV-vis spectroscopy, infrared spectroscopy, or IR for short, NMR, which stands for nuclear magnetic resonance. And that one gives us all kinds of information about the structure of the molecule. And then there's mass spectrometry, which is hinted at in pharmaceutical analysis. These techniques basically use different types of light or magnetic fields to identify and measure different substances based on how they interact.
Okay.
It's kind of like giving each substance a unique fingerprint.
That's incredible. I mean, the level of detail and precision they can achieve is mind-boggling.
It is. But sometimes those good old-fashioned methods are still really important.
Oh, really? Like what?
Like titration, for example.
Yeah.
That's a classic technique that's been used for ages to figure out the exact concentration of a solution.
Okay.
And then there are all those physical tests we mentioned earlier, like for tablets. There's the dissolution test, hardness test, and something called a friability test, which basically measures how easily a tablet will crumble.
So think of everything.
They try to.
Yeah.
All of these tests together, all these different techniques, give a really complete picture of whether a drug is good to go, before it ever reaches a patient.
So that product release testing is really like that final safeguard to make sure that every medicine that makes it out there meets the strictest standards. But once it's made and it passes all those tests, how do we know it's going to stay effective over time? Doesn't it degrade or go bad eventually?
Well, that's where stability assessment comes in.
Okay.
So stability testing is all about figuring out how a drug changes over time and under different conditions. Think about it. Medicines can be exposed to all kinds of environments. It might get too hot or too cold, too humid, or even be exposed to sunlight.
Right.
So stability testing tries to mimic all those different conditions, but in a controlled way.
So they're basically putting the drugs through like a science-backed version of accelerated aging.
Kind of, yeah. So they store the drug under all these different stress conditions, harsher than normal, and also under those ideal conditions, the ones you see on the label, like store at room temperature for long periods of time.
Okay.
And then at set times, maybe every few months or so, they pull out samples and test them using those same analytical methods we talked about earlier.
And that's how they know how long a medicine will last, right?
Exactly.
Yeah.
The stability tests help determine the shelf life, which is that expiration date you see on the bottle, and they figure out the best way to store it too. Like, does it need to be refrigerated or kept away from light? Things like that.
That makes sense.
Oh, and it's worth mentioning that there are internationally agreed upon guidelines for this whole stability testing thing. Like the ICH guidelines mentioned in Season 3, the process of new drug discovery and development, and Season 5, safety evaluation of pharmaceuticals and medical devices international regulatory guidelines.
Mm-hmm.
ICH stands for the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use.
That's a mouthful.
It is, but basically it means that scientists all over the world try to follow the same rules for drug development and quality.
That's good to know. So it sounds like there's like a very well-defined rule book for all of this. But who exactly sets these standards for analytical methods and all the testing?
Well, in the U.S., a lot of it comes down to the FDA.
The Food and Drug Administration.
Yep, and other countries have their own regulatory agencies too. These agencies are, responsible for public health, so they have very, very strict rules for how analytical methods are used in drug testing. They don't mess around.
I bet. So one thing you mentioned was this process called validation in the context of these analytical methods. What exactly does that mean?
So method validation is all about proving that a specific testing method does what it's supposed to do.
Okay.
It has to produce results that are accurate and that can be reproduced, like every time.
Right.
And that means proving several things. First, accuracy, meaning that the method actually gives you a result that's close to the real value.
So, like, if there are actually 500 milligrams in that pill, the test better say it's close to 500 milligrams.
Right. Then there's precision, which means if you test the same sample over and over, you should get pretty much the same result every time.
So it's like hitting the bullseye every time is accuracy, and then precision is like all the arrows being really close together.
Yeah,
Yeah.
that's a good way to put it.
Yeah.
And both are crucial because if a drug is inaccurate, meaning it's consistently giving the wrong dose, or if it's imprecise and varies a lot from dose to dose, that could have serious consequences for patients, right?
Absolutely.
And then there's specificity, which means that the test can accurately measure just the thing you're interested in without being thrown off by anything else that might be in the mixture.
Okay.
And sensitivity, which is super important for drugs that are given in low doses, as mentioned in Season 7 Formulation and Analytical Development for low-dose oral drug products.
Right.
Because in those cases, you need to be able to detect even tiny, tiny amounts of impurities.
Got it.
And lastly, there's robustness, which means that the method still works, even if there are slight changes in the conditions.
Okay.
So it's not enough to just have a test that seems to work. You got to prove it.
You got to prove it.
Prove it with solid evidence that it's reliable and it's going to give you the right answer every time. And all of this is regulated by those good manufacturing practices, or GMP, which are highlighted in Season 6, the Certified Pharmaceutical GMP Professional Handbook. GMP regulations are basically the rule book for making medicines.
Okay.
And they require the use of these validated analytical methods, and they require super detailed documentation of every single step of the testing process. Everything has to be recorded and traceable, no cutting corners.
So there's no room for error.
Not really.
And then there are also those pharmacopoeias like the European pharmacopoeia, the Japanese pharmacopoeia, and the United States pharmacopoeia mentioned in Season 8, merged HU48. What are those exactly?
So those are like official books, essentially, that lay out standard methods for testing lots of different drugs and substances. And they provide a kind of, a baseline for quality testing. And there's this initiative called ICHQ4B that's trying to make sure these pharmacopoeias are all compatible with each other, which would make things a lot easier for everyone, especially for companies that want to sell their drugs in different countries.
It's really incredible when you think about it, all the layers of checks and balances that are in place to ensure the quality of our medicines. But I'm guessing that developing these really robust and reliable analytical methods is no easy feat, especially with all the complex drugs out there these days.
Oh, you are absolutely right. It's a huge challenge, especially when you're dealing with formulations that are really complex, like, combination drugs that have several active ingredients in them, or when you have a totally new drug that no one's ever tested before.
So, like, what kinds of challenges do scientists face when they're trying to develop these methods?
Well, selectivity is a big one.
Selectivity.
Yeah. So remember how we talked about how a test has to be able to pick out the specific ingredient you're interested in without getting confused by anything else that's in there? Well, that can be really tricky, especially when you've got all these other inactive ingredients in the mix, what we call excipients. Okay.
Mm-hmm.
And then, of course, there might be some degradation products forming over time, too. So you got to make sure your test can ignore all that stuff and just focus on the active ingredient.
Right. So, like, separating the wheat from the chaff.
Exactly. And then there's sensitivity, which we mentioned before. If you're working with a drug that's given in very small doses, you need a test that can pick up even the tiniest amounts of impurities. So that could be a big hurdle, too.
Okay.
And then there's the stability of the analyte itself.
A analyte.
Yeah, that's just the fancy scientific term for the thing you're trying to measure.
Right.
So some analytes can be quite unstable once they're in a solution. Like they might start to break down before you can even measure them.
Oh, that doesn't sound good.
Not at all. If that happens, your results are going to be all over the place.
Right. So what can scientists do to get around these challenges?
Well, it often starts with a lot of trial and error, honestly. They have to tweak and refine the testing method until they get it just right.
Okay.
And luckily, there are always new and better analytical techniques being developed. Technology is constantly evolving, which helps a lot. And of course, once they think they have a good method, they have to validate it, remember.
Oh, I got to prove it works.
Exactly. They have to show that it's accurate, precise, specific, sensitive, and robust. But the work doesn't stop there. Even after a method's been validated and is being used routinely, they have to keep monitoring it just to make sure it's still performing as expected.
It seems like a lot of work, but it's clearly essential. It's amazing to see how much goes into ensuring the quality of our medicines, and it's not just about testing the final product, is it? I know from some of the other stuff we've been looking at that these analytical methods are used much earlier in the drug development process, too.
Absolutely. They're really crucial throughout the entire process, from the very early stages of discovery all the way through to post-market surveillance. For example, in those preclinical studies, the ones that are done in animals before a drug is ever tested in humans, analytical methods are used to measure how much drug is in the animal's blood or tissues.
'Cause so they're figuring out, like, how the drug moves through the body.
Right, how it's absorbed, distributed, metabolized, and excreted. We call that pharmacokinetics, which we discussed in Season 2 Basic Pharmacokinetics and Season 3 Basic Pharmacokinetics.
Okay.
And then later on, when scientists are trying to figure out the best way to formulate a drug, like whether to make it into a pill, a capsule, an injection, or something else, they use analytical methods to test things like the solubility of the drug, which is discussed in Season 6 Handbook of Solubility Data for Pharmaceuticals. And then, of course, they have to test how stable those different formulations are.
Right.
And even in clinical trials, when a new drug is being tested in people, they use these analytical methods to measure the drug levels in the patient's bodies, which helps them understand how the drug is working and whether it's safe and effective. So it's really a continuous process.
Wow, that's fascinating. It really highlights how vital analytical methods are to the entire pharmaceutical industry and ultimately to public health. I mean, every time someone takes a medicine and it works, it's partly because of all this incredible scientific work that's happening behind the scenes. These methods are the unsung heroes of drug safety and efficacy.
I couldn't agree more. They're the guardians, working tirelessly to make sure that all those medicines out there are consistently safe, effective, and top-notch quality.
So let's wrap things up with some key takeaways for our listeners. What are the most important points you want them to remember?
Okay, so the big picture is that analytical methods are absolutely essential for making sure that all drug products, everything from pills to injections to creams, have the right identity, the right strength, the right quality, and are free from harmful impurities.
Mm-hmm.
And this isn't just a one-time check at the factory. It's an ongoing process that starts when a drug is first made and continues throughout its shelf life.
Okay.
Those strict industry standards and the oversight from those regulatory agencies, plus all the work that scientists are doing to develop better and better analytical methods, all of that comes together to protect patients and make sure that the medicines they rely on are safe and effective.
And that brings us to our final thought for you, our listener. Think about all those incredible new medicines that are being developed these days, things like biologics and nanomedicines, which are mentioned in Season 3 Pharmacokinetics and Pharmacodynamics of Biotech Drugs, and Season 7 Nanoparticles for Drug Delivery. These are incredibly complex therapies, and they're pushing the boundaries of what's possible in medicine. But how do you think those analytical methods we've been talking about today are being adapted to make sure these new treatments are safe and effective? How do you ensure the quality of something that's so cutting edge and so intricate?
It's definitely something to ponder, isn't it? Absolutely. It's a huge challenge, but it's one that scientists are tackling head on. And if you're interested in learning more about this fascinating field, there are tons of resources out there, like from the FDA or the ICH. But for now, we'll leave you with this question. What do you think is the single most important aspect of ensuring drug quality through analytical testing? We'd love to hear your thoughts.
Thanks for joining us on this deep dive. We'll see you next time.